Herring was interested in diphtheria, and then the 1800s and the early 1900s, this is a disease that killed lots of children. Go to graveyards from that time and you'll see lots of children between 1 and 5 that were killed by this disease. So he experiments with guinea pigs and injects the toxin from diphtheria into the guinea pigs that have recovered from diphtheria, and they don't become ill. So something is protecting them against this toxin. He comes up with the idea it must be an antitoxin. So this morning I was saying, it sounds like Star Trek. Remember Star Trek? It's like there's always a point in Star Trek where he goes, "Captain, you can't mix matter and antimatter." Remember that? Scotty, the engineer. If anybody watches Star Trek anymore. A few students this morning did. Anyway, something called, we put in a toxin, so something must be neutralizing it called antitoxin. And so then the other thing he did is mix toxin with serum from a guinea pig that had survived, assuming it had the antitoxin in that serum, injected it into another guinea pig who hadn't been exposed to the toxin, and it remains well. So we have this idea there's some way to create specific immunity against certain diseases. The first antitoxin was given to a child in 1891, so now we have a cure for children that have diphtheria. He wins the Nobel Prize in 1901 for this work. Okay, so what does this have to do with diphtheria toxin? My students were totally stumped this morning on this, so I'm relying on this reminding you of something you might have seen. So if you watch a lot of Disney movies, you might...
Is this is that Balto?
Balto, of course it's Balto. Yes, and how do you know Balto?
It's the dog that ran the last leg of getting the medicine to an Alaskan
Right.
village or whatever.
Right. Right.
Thank you.
And how do you know that? Did you watch the Disney movie?
I've watched a Disney movie, and actually recently I read a post about another dog, Togo, that ran the longest of all of them. So
you're a dog person?
Yeah.
Okay.
But,
Okay.
Enough.
I see. So you know where Balto is now?
No, I don't.
He's at the Cleveland Zoo. Obviously, he's stuffed,
Yeah.
but you can visit Balto. And if you go to New York, by the Children's Museum, there is a bronze statue of Balto in the ground. So that's how famous this dog was. So, in case you guys are just studying all the time and not clued into all this sort of immunological trivia and disease outbreaks. So there's an outbreak of diphtheria in Nome, Alaska in January 1925. The closest antitoxin was in Nenana, which is 675 miles away. Now, I don't know whether you've been to Alaska, but in January, especially at that time, with the equipment we had, you couldn't fly a plane, right? It's just way too dangerous. So the solution is you set up a relay of 20 mushers, and what they're going to do is they're going to be at each stage along the way to get this antitoxin to Nome, and it's going to be passed on from one musher to the other. This took them 5.5 days to accomplish this. It is the basis of the Iditarod sled race, in case you've heard of that. They only actually, I think they only go a couple of, about half of this route that they did in 1925. It's also the Great Race of Mercy. So at the time, it was like the biggest thing, you know. I mean, who doesn't want this heroic act? You have, children that are sick 600 miles away and you have these heroic people leading on the, passing this antitoxin from one to the other, round the clock, 5.5 days. So this is Balto, which was the lead dog on the last, well, the second last and the last. Maybe you know that. So this is a Norwegian, Gunnar Clausen, and he gets to the point where he's supposed to hand over the toxin for the last leg, and the guy's like asleep, and his dogs are not ready, so he goes, hell with that. We're going for it. And so he brings it in with Balto, and so both of them became like big celebrities. Like they were at Madison Square Gardens. People just wanted to see them. So this is how cool microbiology can be. Heroic people and delivering, especially if you're delivering aid to children, it's even more captures the public eye. So, any other questions on that, or is that anybody going? Oh yeah, I think I heard this before. No. Okay, so you're the lone person in this class. So let's get into some immunology and starting to understand these different types of cells. So lymphocytes, you're going to find those in the lymph system. And remember, they are also leukocytes, but they're one group of leukocytes, and specifically the lymphocytes. So there's 3 different classes you can put them in. They can be immature lymphocytes. They haven't properly matured. And so they don't have the receptors that they need. You can have naive lymphocytes. These are lymphocytes that are mature but have never encountered the antigen that they're programmed for. And then you can have activator lymphocytes. These are being activated by binding to the antigen that they're designed to bind to. And we can talk about them as being effector lymphocytes. In other words, these are the ones that are going to be involved in fighting off the disease at that time. And then some of them hang around, and these are the memory lymphocytes. And this is just following that little cartoon that you saw when I showed you the video about vaccines. So we have ones that are dealing with the infection, and then ones that hang around after the infection's over. So you might ask, how does this happen? How can they possibly be sensitive to different antigens? Both of them, both the B cells and the T cells, these are the lymphocytes can be broken into B cells and T cells. Both of them have a receptor on the surface. B cells is a little easier to understand, so think of your B cell, and what it has is the antibody it's going to produce sticking out of it. And so when the antigen comes along and binds specifically to that, remember there are like 1 billion B cells and T cells, they only will bind to one specific antigen. So when that antigen comes along, it binds, and then that's going to trigger a response. And so you can see that makes a lot of sense. Then through a series of steps, it's going to start to make that same antibody, and then that's going to be in the bloodstream and go into the tissues. And so that's going to be able to bind up this specific antigen. T cell receptors, they don't recognize the free antigen. So this one, the B cell, the antigen floating around, it'll bind to it. The T cells don't recognize a free antigen. They have a different type of receptor, which is it looks like 1/2 of the arm of an antibody. What has to happen is these are going to be presented with the antigen by another cell. And so here's the T cell receptor, and then here's another cell that's going to actually present it to it. And so that makes it a little more complicated, but what you're going to understand is all these cells are cooperating with each other. And Andrew, if you could just get off your phone, would be helpful. Because if you're not paying attention and taking notes, you will spend a lot more time at home trying to understand all this. So, everybody get this. There's they both have receptors. They have different types of receptors. This one looks like the antibody they're going to produce. This one, they have a receptor, but the antigen has to be presented to it. So where you're going to find lymphocytes? Obviously in the lymphatic system. So this is a system that runs through your body, and what you have is blood going through capillaries. Some of the lymph or the plasma is going to leak out into the tissues, provide the tissues with nourishment. But it doesn't go back into the capillaries. It's actually going to be collected by the lymphatic system. So this is all the lymph is draining back into the lymphatic system, and then it eventually ends up back in the blood. So what the lymphatic system is, it's sort of like, if you want to catch a crook, have the cop hang out on the highway and look for them coming along. And so what you have is these highways of fluid from the tissues, and that have to pass by the lymphocytes. And so they're going to just check, is everything okay there? So what you negate is having to have T cells and B cells down in your toes. You don't need that. You just need that fluid draining back, and then it being surveilled by the lymphocytes. Everybody good with that? So these are the primary organs of the lymphatic system. The secondary ones are going to be things like the lymph nodes, the spleen, the tonsils. So when you go to the doctor and you say, "Doctor, I'm not feeling well, I'm lethargic, I think I got a slight fever and things," what's the first thing they do?
Check your lymph nodes.
Right. What they're looking for is, are your lymph nodes inflamed, indicating you've got some sort of an infection going on. It's a cheap, easy way to figure out if somebody's got something going on, or there's some other issue with them. But so when you have an infection, then the cells in the lymph are going to start multiplying, and you are going to get a little tired if you're mounting an immune response. You've also got mucosal associated lymphoid tissue, MALT, which prevents microbial invasion through the mucous membrane. So here, for example, the intestine, and you have antigens, and you have macrophages and dendritic cells sampling what's going on in the intestine, and also preventing any invasion of microbes into the body tissues. In your skin, you have something called SALT, skin-associated lymphoid tissue. Again, same sort of system where you have dendritic cells, macrophages, checking to see if nothing's invading.
So is lymph considered like a fever maker, or no?
Lymph itself is just a fluid. Right. Lymph is the liquid that drains from your tissues.
So bacteria causing our lymphocytes as well.
Right.
Lymph nodes as well.
Right. It's the bacteria interacting with your T and B cells and your macrophages to cause it to swell in that particular area. and you're going to have proliferation of those cells, and so something is going on. That's why you're going to be able to feel in your lymph nodes a swelling.
So is the bacteria considered the pyrogen?
A pyrogen would be, it's considered the antigen. A pyrogen would be specifically a part of a bacteria like lipopolysaccharide that causes you to get fever. Anybody else? There's lots of these terms, and I know it's confusing. So if you start to get into a position where you're like confused, don't be afraid to just say, "Whoa, let's go over that one more time." So what we're talking about today is adaptive immunity. What we talked about on Tuesday was the immunity that generally keeps bugs out and generally recognizes microbes. Or recognizes, so if you know, you get injected by a microbe, then or infected by a microbe, then in the innate system, it's just going to go, oh, there's peptidoglycan around. That's a problem. I recognize that as a problem. However, if in this system, it's going to recognize specific bacteria or viruses. And so that's the thing about it. It has specificity. It also has a memory. So when you get, if you get vaccinated for SARS-CoV and then you actually get the disease, then you've already built up memory cells, so you have a strong response the second time you see it. So that's the beauty of it, specificity and memory. However, this takes a while to build. And so if you're feeling lucky, as Clint Eastwood used to say, you can roll the dice and say, I've got a great immune system. If I get COVID, I'll be fine. Now, there's 1.1 million people who rolled the dice that are not here anymore. So I don't advise you try this one with COVID, because what happens is this system kicks in, okay, but it takes, a week or more to build up. Now, in the meantime, you're just relying on innate immunity, and that may not be enough. So the bug may kill you before your immune system kicks in. And so that's why, vaccination is important. It's like, well, maybe you would have been okay had you not been vaccinated. But do you want to take that risk on any of these diseases? And because, it's not a video game you're playing. There's no like, oh, let's reset. No, you are not here anymore to reset. Sorry to make it so dramatic to you guys, but so when you get exposed to an antigen, and we'll talk about what an antigen is in a few minutes, when you get exposed, you have a primary response, and the adaptive system remembers that antigen. When it encounters it again, we have what's called a secondary response. The secondary response is always stronger. So instead of letting you catch the disease and saying, well, you should probably not catch it or you get a milder case, what we're encouraging in the health system is you get vaccinated first. That's your primary response, and then when you encounter it again, the body is able in a day or so able to sort of mount a strong defense against that pathogen. So there's two strategies in the adaptive system. The humoral immunity, where we're going to eliminate extracellular antigens. So we're going to eliminate bits and pieces that we find, either the whole bacteria or pieces of the bacteria or toxins. This is humoral, involves antibodies. So if you can remember that, that's helpful. It also, the humoral is associated with the B cells. So here's some connections I'm trying to make in your brain. Cell-mediated immunity, or cellular immunity, this is dealing with antigens residing on cells. So if one of your cells in your body is infected by a virus, the T cells can detect that, and then they will line up alongside it and bombard it with toxic chemicals and kill that cell. So it's better to kill one of your cells, have your immune system kill one of your cells, and then stop the replication of that virus than let the virus continue multiplying in there. Everybody good at this point? And all of these things like build on each other. So we're going to give you some of the basics and then just go a little deeper. So the big picture for humoral immunity is we have B lymphocytes, cells that reside in the lymph system, or they're also called just B cells. They develop in the bone marrow. So you remember B cell, bone marrow. You've already answered a question on a quiz. And so they are triggered to proliferate when they're exposed to an extracellular antigen, an antigen that's floating around. They differentiate into plasma cells. So the plasma cells are the ones that are going to produce the antibody. How you trigger this? Through that B cell receptor. That's going to trigger the response. However, they don't just do it on their own. What's going to happen is they need a T cell to confirm that that antigen is something you respond to. And one thing, you know, it's maybe not the best example, but you know, they talk about the government having checks and balances to it. American democracy, checks and balances. That's why you can often not get anything done. But in the immune system, you don't want immune cells just going off on their own. So with the B cell, it responds to the antigen, but that needs oftentimes to be confirmed by an actual T cell. So it's not just doing its own thing. Antibodies are then going to protect by binding to those antigens, binding to those bacteria, binding to that toxin, binding to that viral cell, and eliminate it from the body by tagging it and making it not active anymore. So antigen, it's always hard to remember what's an antigen and what's an antibody. Well, an antibody is produced by the cell in the body. An antigen means an antibody generated. So that's where the term comes from, and that might help you keep those two things clear. An antigen that elicits an immune response can also be called an immunogen. And this is how in immunology and medicine you end up giving things two different names. So, in general, the antigens come in, you can classify them in two ways. Most are T-dependent. In other words, you need a T cell to help the B cell activate itself to produce the antibody. You also have T-independent antigens that activate the B cells without a T helper cell being present. Things like lipopolysaccharide will do this. So when you're trying to get an immune response, proteins are great for producing an immune response. The weaker are lipids, and small molecules just aren't going to elicit immune response. So you need a big molecule that has, going to persist and has lots of sites. The site that the antibody is going to recognize is called an epitope. So when I say that so here's our protein, whatever it's doing. it's not the whole protein that's recognized. It is only a part of the protein. This is called the epitope. And this is what, the antibody is actually going to bind to a part of the protein. So we call that the epitope. So what the antibodies look like, they look like Ys. They have a FAB region and FC region. The FC region is involved in complement fixation. Here are the active sites at the end of the Y. So this is a molecule, with its hands right out, ready to grab onto something that it recognizes. It has 4 chains. 2 of them are what we call heavy, or they're larger chains, and then 2 of them are small. At the end of the chains, so these ones are constant up to this point, but at the ends are the variable regions. As the cells develop, then the antibodies they produce are going to be different. The amino acids are going to be different. And this is all part of processing introns and exons. You've heard about that. What these are doing are processing those deliberately differently, so you have a lot of variation in what these cells have at the antigen binding site. And so you can respond to about 1 billion different epitopes. And the reason for that is because this region is variable from one cell to the next, and it gives it a particular specificity. Everybody good with what an antibody is? So, why are they so magic? Well, they do all these different things. So, if you have a virus, say you have the SARS-CoV virus, and you've either seen it before, or you've been vaccinated, then the antibody will bind to the surface of that. If you bind to the surface of it, then you neutralize it because it can't cause any more damage.
I think those kind of they make someone clump together almost like they can move more.
Sorry?
Like anta, like kind of like bind so you can like clump together, like
yeah.
you can't move.
Yeah, well, a virus couldn't move anyway, but what it wants to do is find one of your cells and bind to it. If we've already coated it in antibody, then you've neutralized it. Same with a toxin, the diphtheria toxin we just talking about. If you bind antibody to it, no longer can it get inside the cells and wreak havoc. So that's just one example of what they do. The other thing is they can coat a bacterium. So its antibody is being produced against something on the cell wall. It coats it, and then that makes it more delicious to macrophages. So it activates the macrophage to take it up and digest it and break it down. You can also have, remember the classical complement activation pathway? So in that, you're going to see complement binding to this outside region, the FC region. And so then you're going to have inflammatory response that's going to be able to lyse foreign cells. You also have, if they're mobile bacteria and the antibody binds to the flagella, then you're going to slow them down. They're not going to be able to move around anymore. So you've immobilized them, so it's like, you put, what are those things in the Texas boot you put on a car, when they find you parking, they kind of clamp your wheel. So that's what you've done here, and then you've got a little more time for the macrophages to come along and eat these up. The other thing you can do is you can cross-link them and clump them all together. That means they're easier to, sort of sweep up. So it's like sort of aggregating these in one place, and so a macrophage can come along and get a big mouthful of these. The last one, antibody-dependent cellular toxicity. This is where you have an infected self-cell. So maybe there's an antigen on the surface that's novel, and it's being recognized, so we're producing antibodies against it. And so this is one of your cells that's infected, and we know it because it's displaying something different on the outside. That's going to attract the natural killer cell, which is going to deliver toxins and enzymes that are going to cause the death of that cell. Everybody see what's going on with this, how this works? That's why people are talking about, oh, antibody levels, and when you get vaccinated, what's the antibody levels? Oh, they're dropping off.
Is this for both T-dependent and T-independent?
Right.
Okay.
Right. Well, we're just talking, yeah, they would both be producing antibodies. It's just the mechanism of how that you get the whole process started.
Oh, I meant, sorry, I got the two of them confused. Is this for both humoral and cell-mediated?
This is just humoral.
Okay.
Because humoral is all antibodies.
All right.
But, I mean, the T cells are helping the B cells.
Yeah.
And we'll talk about that in a minute. So you have different classes of antibodies. When I was a student, just a few years ago, we remembered this as Gandhi. I can't remember. I don't think the movie Gandhi was out at that point. But anyway, so G-A-N-D-E. That's how we remember the different classes. They are a little different because here's the first class that's produced when you have an infection is IgM. What you can see that is a pentamer, so there's 10 different sites for binding on this particular antibody. IgG is the most abundant that you find, and it's the most abundant after in the secondary response, and it also activates complement. So there's IgM too. IgA is what you're going to find in mucosal tissues. So it's secreted in saliva, tears, and mucus. IgD is involved in the development and maturation of the response. IgE is one that's involved in allergic reactions. So what this one will do, and I'm telling you something from the next time we talk. So if you have a mast cell, they're full of granules of histamine. Along comes, what they will do is they will bind the antibodies on their surface. So now they're like a time bomb, So along comes, your pollen grain. And once it binds to once it binds to the antibody, then it will cause degranulation, and then you'll have histamine, sneezing and coughing and whatever else that go along with it. So you set up the mast cell to be very sensitive to that particular antigen, and then you're going to have a very dramatic response. We'll talk about that because the next lecture, next Thursday, is going to be about what happens when this goes wrong. Because, as you can see, powerful system, but you don't want to get it getting out of control. Yeah?
So is that also, you're talking about sneezing and stuff, but is that also with an allergic reaction? Like how that works? Okay.
Yeah.
And then we're going to go into autoimmune or something?
Yeah, yes, exactly. You can't wait, can you? So the other thing I want to make clear is how this works. If you are exposed to an antigen, somewhere in your body there is a cell that's sensitive to it. What you're not doing is setting off all the cells at once. It's not every B cell starts to proliferate, because why would they? They're producing antibodies you don't need. What happens here, and I do seem to This is fuzzier than I thought. Anyway, here we have the B cells being produced, and every one is sensitive to will bind a different type of antigen. So you have this great differentiation and diversity of these cells. However, along comes an antigen and it binds to this particular B cell because it has the receptor on the surface is designed to just bind to this one. What happens then is you have activation of that B cell, and you have multiplication of it, and then you have more proliferation and differentiation into the memory cells that are going to hang around, and then also the plasma cells that are going to produce the antibody as long as the antigen is around. And so that's what happens. It's not every one of your B cells takes off when it sees the antigen. It's only one of those. And then they're going to proliferate places like your lymph nodes, and you're going to have a production of this. It's going to end up in the bloodstream, and so that's the way that this occurs. Everybody good with that?
So it basically assigns the specific one specific for that task whenever it recognizes the antigen.
You randomly have say 1 billion B cells, and then when that antigen binds to it, that's what sets it off. It only sets that one cell off, though, and that's going to proliferate and produce your memory cells and your plasma cells. So let's just, you've got a lot of the basics here. I'll just go through a little bit more of humoral immunity so you understand it. So what starts the process is the B cell has a receptor that binds to the antigen. The antigen, though, it's not just the fact that it binds to the antibody like receptor on the surface. It is then internalized and degraded into peptide fragments. The fragments are delivered to the MHC, class 2 molecules, for inspection by the T helper cell. Okay, so it's binding to that receptor, then it's internalized, broken down, bits of it are shown to the outside to the T helper cell. And that's called antigen presentation. If the T cell receptor, remember, we talked about that those antigens have to be presented. If the T cell receptor binds to it, then the T cell activates the B cell with cytokines and starts this process of proliferation. However, if no T cells recognize the peptides, then the B cell becomes anergic. In other words, you tolerate that particular antigen. You're not going to mount an immune response to it. And so what I just said, this is basically the picture version. And so here's our B cell. Remember, the receptor looks like an antibody, binds to an antigen that is specific for that particular receptor. And then you're going to have internalization. You're going to have the antigen broken down. And then it's going to be displayed on the surface as a fragment on the major histocompatibility class 2 receptor. So there's two ways it can go. If it's presented, if the T cell binds to it, the original molecule broken down, presented on the surface, if it binds to it, then we're going to have activation of this B cell via cytokines that are going to tell this cell to start proliferating. However, if the T cell doesn't bind to the fragments on the surface, then you have no proliferation, and basically you're tolerant of that particular antigen.
Can you relate the terms anergic and immunogen?
Well, anergic just means you're tolerant. At this point, there's no reaction. An immunogen is a antigen that causes you to produce an immune response, which in this case would be antibodies. And to produce the antibodies, in this case, because it's a T-cell dependent antigen, then you need the OK from the T-cell to activate the B-cell. Everybody, does that answer your question?
Yeah, so energetic refers to, like, that could be used to define an antibody or like characterize an antibody, but immunogen refers to the antigen.
Well, yeah, the body would be, the B cell would be anergic. In other words, it doesn't react. So it finds something foreign and it just, nothing's going to happen in this case. In this case, find something foreign, T cell says, holy, you know, you better start proliferating because, you know, that's something I've recognized before and it's recognized as dangerous.
And if that B cell didn't have that specific receptor for the antigen, then we don't go to step 4, step 5A, or step 5B.
You need this particular cell needs the receptor that will bind this particular antigen.
Okay.
Yeah. So getting back to the immune response, you have 10 to 14 days before you see substantial antibody accumulation. In that time, you're on your own trying to fight off this infection. And so you've got the differentiation of the B cells into plasma cells, and they're going to generate the antibodies. They're going to start to die off unless you have the antibody continually in your system. And so what you see, the first antibody produced, it's going to be IgM and then IgG. However, the second response, so we just talked about the primary response, the second response, you see the IgG being the most common, or the most frequent, or most IgG is being produced. IgM, the same sort of a response. But you can see the advantage of the secondary response, so you can see why we vaccinate. You get a response, but it's not very good. So you either get this from being exposed to the pathogen or being vaccinated, and then the secondary response is much faster and much stronger. And so that's the science behind vaccinations. And once you've had the primary response, you have those memory cells. They're just waiting, and they can proliferate much faster than if you have to start the process from the beginning. Everybody good with that? So those are T-cell dependent antigens. We also have T-cell independent ones. These are things like lipopolysaccharides. What they can do, because they're long repeating molecules, they can stimulate lots of the receptors here on the B cell, and so they don't need a T cell to come along. They can, so that's why you end up with a T independent antigen. Okay, anything on the humoral immune system that's not quite as clear as you would like it?
What is done with the waste from all of the destroyed cells?
So the waste, I mean, things like macrophages will sop up sort of waste and break it down. I mean, it ends up being solubilized, and then you'd excrete it in your kidneys.